Good morning, everyone. Magandang umaga sa ating lahat. I'm Angela Melissa Carlos, PSM Board Representative for Microbiology Education and Treasurer of the Microbiology Council. I will serve as your host and moderator for this morning's program. The proceedings today would feature the launching of two MCC projects, as well as the presentation of remote learning materials for basic microbiology laboratory classes. Materials will be made available to all member HEIs of the consortium. To open our event and welcome all of you, let me call on the President of the Philippine Society for Microbiology, Professor Joel C. Cornista.
Good morning, everyone. On behalf of the members of the National Board of the Philippine Society for Microbiology, I would like to welcome you all to the Microbiology Consortium Council launching of the OBE monograph. After a series of workshops conducted on outcomes-based education in microbiology with different member universities all throughout the Philippines, a copy of the monograph is now available for distribution to consortium member HEIs. I would like to thank the National Academy of Science and Technology for jointly publishing this monograph with PSM. MCC will also be launching today their website where learning resource materials for microbiology, such as harmonized syllabi, blended laboratory exercises, and other important materials and information for microbiologists will be posted. These resources would definitely be very helpful in microbiology professors handling microbiology subjects from different universities throughout the country. While some of the resources are open to all, some are limited to members only, so we therefore encourage everyone to become a member of this microbiology consortium. Also happening today is a webinar on the development of remote learning modules for the laboratory component of the basic microbiology course which will be presented by different member universities such as the University of the Philippines, Manila, UP Los Baños, UP Diliman, UP Visayas, Mindanao State University, IIT, Cavite State University, Mariano Marcos State University. This will also be very significant, particularly in this time of the pandemic, university offering microbiology courses online in a blended mode. Indeed, the Microbiology Consortium has come a long way, starting as a committee to the Microbiology Education Division of PSM to what it is now. Of course, I would like to thank the head of the consortium, Dr. Asuncion Raimundo, for making all of this possible. Especially thanks to the chair of the Microbiology Education Division, Professor Angela Melissa A. Carlos, and to the other hardworking members of the MCC board, such as Dr. Wendell Rivera, Dr. Bernadette Mendoza, Dr. Rex Sadava, Dr. Franco Teves, Dr. Prima Franco, Yolanda Ilagan, and Bondato for their dedication. Again, thank you very much for attending this launching and webinar of MCC, and we look forward for more projects and workshops of MCC which are already in line for this fiscal year. Magandang umaga po sa inyong lahat, and we look forward to this very fruitful launching and webinar. Maraming salamat po sa inyong lahat.
Thank you very much, Mr. President, my president. Now, let me just inform everyone that we will have an open forum at the end of all the presentations. So please type your questions in the chat box. Today, we would see the fruition of a project which had its inception in 2018. To proudly share the finished product with all of us, I'd like to present our beloved MCC Chair, Dr. Asuncion K. Raimundo. Ma'am Shoen.
Oops. Good morning, everyone. I'm glad to see familiar names in the list of participants, and they are mostly members of the Microbiology Consortium. It has been said that teaching is not a profession, but a passion. It must have been passion that drove me and the authors to finish this monograph. It took 2 years and a pandemic to allow some of the authors to finish their share of articles. Finally, all efforts came to fruition, and now this monograph titled Outcome-Based Education, OBE in Microbiology, is now in print. And this is through the support of the National Academy of Science and Technology and the Philippine Society for Microbiology. Is this OBE monograph still relevant at this time of pandemic and thus remote learning? Very much so. Learning outcomes have still to be considered. Teaching materials must be learner-centered, and all the other principles of OBE have still to be considered. This is the monograph, and I'd like to share with you the contents of this monograph. We started with an article on expanding microbiology workforce in the Philippines, the birth of the Microbiology Consortium. I thought it proper that we should put in writing how this Microbiology Consortium came into being. Next article is from Dr. Esperanza Cabrera from the La Salle University, and she discussed what is outcomes-based education. Next article is from Professor Rosemary Sabio from UPLB, University of the Philippines, Los Baños. Originally, she was also writing about the principles of outcome-based education, and she had the hard task of shifting gears, and had to write on authentic assessment and outcomes-based education, a perfect match. The next article is by Dr. Franco Teves, and in his article, he shared with us his experience on how the Mindanao State University Iligan Institute of Technology went through the process of converting their curriculum, their courses, into outcomes-based education mode. Dr. Thomas Edison E. de la Cruz from University of Santo Tomas wrote an article on learning is fun, developing outcomes-based, engaging learning activities in microbiology. You should be able to pick up a lot from him from this article. The next article is written by Dr. Bernadette Mendoza of University of the Philippines, Los Baños. And she's sharing with us her tips and tools on how she teaches microbiology the OBA way. Then we solicited an article from Dr. Windel Rivera of UP Diliman. We solicited his views on the BS Biology program with specialization microbiology as recommended by CHED, as written in the CHED Order Memorandum Order 49 series of 2017. So you will see there the thoughts of Dr. Windel, Dr. Rivera on this program as stated in the Memorandum Order of CHED. And last but not the least, is Dr. Pawilan already in, is a sort of summary of all the articles, designing outcomes-based education for higher education institutions, weaving the experience of developing and implementing microbiology curriculum in the Philippines. Dr. Pawilan had three roles in this monograph. When I was organizing this monograph, I was aware I know my microbiology, but I'm no expert on OBE. So I sought a person who is knowledgeable on OBE and in curriculum development. And that led me to Dr. Greg Pawilan. I asked him to review the articles. Eventually, I asked him to be a co-editor, and eventually he wrote this article. I would also like to acknowledge the people who contributed a lot in the fulfillment of this monograph. Of course, the PSM and NAST are foremost in the support in the publication of this monograph. And then the different chapter heads when the series of OBE workshops were held. Dr. Janet Supremo was the president then of PSM Mindanao chapter. Dr. Doralinda Lisa was the president of PSM Visayas chapter. Professor May-Ann Batuyong was the president of PSM chapter, Luzon chapter. And the Northern Luzon workshop was organized by Dr. Windel Rivera. I have to mention Dr. Dean Jerome Buenviaje. He was one of the reviewers and he did a good job in recommending improvement of the articles. Dr. Dizon, Dr. Bautista were the editors of this monograph, and Bonnie Bon, who is always around during our webinars, and Prof. Mancilio helped out in the laying out of this monograph. Now I'd like to call on the Executive Director of the National Academy of Science and Technology, and I'd like to hand over a copy of the monograph. Can you spotlight Dr. Prof. Madam Ningning Samarita Bon? There, can you spotlight? There. Madam Ningning Samarita, may I hand over a copy of the monograph?
Thank you.
Okay.
On behalf of NAST.
Okay. Now I'd like to give a copy to each of the authors. Is Prof. Pawilen already in?
No, ma'am.
Is he in? No? Okay. Okay, I'll start then with Dr. Esperanza Cabrera. Spotlight on Dr. Cabrera, please. There she goes. That's Dr. Cabrera. Is your copy? Okay. Next is Dr. Prof. Rosemary Eusebio. Prof. Eusebio, here's your copy. There you go. Thank you. Next is Dr. Franco Teves. Dr. Teves, here's your copy. And there you go. Okay. Then we have Dr. Edison. Is he around? He must be.
No,
okay. So now I call on Dr. Bernadette Mendoza. There. Thank you, Dad. And then Dr. Windel Rivera. Spotlight Dr. Rivera. Where's Dr. Rivera? There you go. There's Dr. Rivera. And Dr. Pawilen is not around. Did not see him. Well, maybe after the video message, he's not around. Okay. Now let us hear a video message from the National Academy of Science and Technology President, Dr. Rodora Azanza.
Everyone, on behalf of the National Academy of Science and Technology, I wish to congratulate Dr. Asuncion Raimundo, academician and Professor Emeritus of the University of the Philippines, and Dr. Greg Pawilen, Associate Professor of the University of the Philippines, Los Baños, for their contribution entitled Outcomes-Based Education, OBE in Microbiology. This is a NAST publication or a NAST monograph number 23. The National Academy of Science and Technology has always been supportive of excellent contributions to the advancement of science in the Philippines. We hope that the universities and colleges in the country and the region would make full use of this book for the development of microbiology as a course and career in the Philippines and the region. During this time of pandemic, microbiology has been considered a very important uh, field in the Philippines and the rest of the world. Virology, which is a field of microbiology, is an important course during this pandemic and should be emphasized in the teaching of biology from the very beginning, meaning even in high school, likewise in the colleges. I wish the authors the editors success in their future endeavors, especially in advancing the field of microbiology in the Philippines. Congratulations also to the Philippine Society for Microbiology for this very significant project. Magandang umaga sa inyong lahat at mabuhay tayo.
Thank you to the National Academy of Science and Technology. I just saw Dr. Greg Pawilen just got in. Can you spotlight on Dr. Pawilen? Dr. Pawilen, I'd like to hand over a copy of the monograph to you. It's good you were able to make it. So here's your copy, and we will send you the actual monograph sometime. Thank you for joining us. Okay. For everyone in the audience, we'd like to inform you that each member HAI is entitled to 2 free copies, one for the library and one for the microbiology unit. But if you want to have a copy for yourself, maybe you can order for a minimal cost. Okay, good morning everyone, and let's move on to the next activity for this morning. Thank you.
Thank you, Ma'am Sean. Marami rin pong salamat sa lahat ng authors sa inyong effort. The pandemic has upended most, if not all aspects of our lives, including the educational landscape. In response to the challenges we're all facing as educators, the MCC has established a website which will serve as a repository of distant educational materials that may be used by consortium members in their respective classes. To give us more details about this very timely and worthwhile project, we have Dr. Bernadette C. Mendoza, Secretary of the Microbiology Consortium Council. Ma'am Det, the screen is yours.
Good morning, everyone. On behalf of the Microbiology Consortium Council, I wish to welcome you to the soft launching of the website of the Microbiology Consortium of the Philippine Society for Microbiology Incorporated. Its creation and development was made possible by the able assistance of Mr. Lee Bolanos. Currently, the administrator of the website is Mr. Boni Datul. The website is actually still a work in progress. So for today, I will just be providing you with some information, bits of information, or shall we say sneak preview on its contents. We will be announcing later more details about our website. Okay, can we go on, Bon? The next slide should show the homepage of the MC website. So let's wait for that slide. So the website will of course show the logo at the top of the microbiology Consortium. So this is the homepage that you're seeing, and below the logo are some tabs to the different website contents. Okay. Next slide, please. For access to the website, this will be disseminated later to the member HEIs of the consortium. Okay. Next slide. Among the contents of the website are the following. So you will see there the origins, information about the start, the beginnings, the origins of the microbiology consortium. Okay. So if we go back to the homepage, this was the picture that you've seen earlier on the homepage. So this picture was taken during the formal launching of the Microbiology Consortium by the MCC chair, Dr. Asuncion K. Raimundo. And this was on the occasion of the 47th Annual Convention and Scientific Meeting of PSM, which was held at Legazpi City in Albay. Okay. So during this event, the heads of Bicol University and the University Result System were there to represent the other HEIs across the country in the signing of the Memorandum of Agreement. On the next slide, okay, so this still contains information about the beginnings of the consortium. So how MC started and embarked on its aim to unite various HEIs in the Philippines towards transforming microbiology education in the Philippines. This information are there in the article by Dr. Asuncion K. Raimundo. Okay. For the other contents of the website, there's also a link to the harmonized syllabi. So what are these harmonized syllabi? These are the harmonized course outlines of 6 microbiology subjects, which will be covered in the licensure exam for registered microbiologists once the microbiology bill is enacted into law. The 6 courses include basic or general microbiology, microbial ecology, microbial physiology, food microbiology, medical microbiology, and industrial microbiology. As a sort of a background on this, workshop spearheaded by the council was held at UP Los Baños sometime in August in the year 2018. So the workshop was on the harmonization of this course syllabi or course outlines. And attending the workshop were representatives from various higher education institutions in the Philippines who were either offering microbiology courses, degree programs, or were interested to offer such. So the main objective of the workshop was to standardize the minimum content of these microbiology courses. So we move on now to another site or another content in the website. This is a very important one, and it's about the links to microbiology learning materials. So this is a compilation by the Microbiology Consortium, and additional resources will be added to this. So in here you will find online educational or learning resources such as the following. So the first item there is B307 Microbiology. This is a Sailor.org Academy free self-paced online learning course in microbiology. So there are also these other contents or links. Biointeractive microbiology classroom resources. What else? Interactive microbiology games, science photo library, virtual microbiology classrooms, and online books. So more will be added. More will be added in time. Next. Okay. Next slide. On next slide are shown other informative articles such as these publications. One is a monograph by Dr. Asuncion K. Raimundo on the assessment of the microbiological needs of the food and pharmaceutical industries. The other one is a journal article on the assessment of microbiology education in the Philippines. Okay, and then another content of the website is about videos or video materials for online, blended, or flexible learning. So, Bon, can you please flash that slide? Okay. So, initially, recorded videos of the webinars organized by the Microbiology Consortium in PSM are posted here. Okay, so two of such are shown. So some of you may have attended these webinars. Later on, video and other online materials developed or created by our very own consortium members or links to such will also be included. Use of such materials will, of course, be subject to copyright considerations. Okay, so for now, that will be all. So it's really just more of a preview. A lot of technical work will still have to be done on the website by Mr. Bolanos and Mr. Datul in cooperation with MCC. Please hope that in about a week's time or so, more information will be added. If you have some technical questions later on the website, we have our technical people around to answer this. Thank you very much.
Thank you so much, Ma'am Det. I'm sure her presentation has sparked a lot of interest in the audience. I would also want to thank once again the PSM for the financial support that they gave us for the establishment of this particular website. The last part of the program would showcase the remote learning materials that I referred to earlier. What you will see, however, is just an overview or a so-called teaser of what each component has to offer. To start off the demonstrations, we have Asuki as our presenter in the person of Professor John Daniel Ong from the University of the Philippines, Los Baños. His team developed a material for exercise number 1 entitled Microscopy. Sir Daniel.
Hello, so good morning to everyone. Thank you for the opportunity. Thank you to the Microbiology Consortium for this opportunity to share our work on this exercise on microscopy. So this materials would not have been possible if not for my colleagues, Dr. Renard Hamora, myself, Picundo, and Professor Michael Nagpala. So again, as Professor Carlos mentioned, this will just be a kind of a sneak peek of what we prepared, because if we go through them completely, then it would take up much of our time. So again, this is part of also the exercise that we are preparing for the basic micro course in UPLB. So exercise 1 is all about microscopy. In UPLB, it's divided into 2, compound microscope and the calibration of the microscope. These are the resources that we prepared. So first, we have the pre-lab video with voiceover, and then a PDF copy of the PowerPoint, and then experiment videos. So just to show you what we prepared. So we have here links to what we prepared. So once you click the link, just screen share this again. Share computers. All right.
Portion of MCB11, Biology and Applications of Microorganisms.
So this is
I am Professor John Daniel P. Ong
voiceover.
from the Microbiology Division of the Institute of Biological Sciences, UPLB.
So just to give you
Under the stage next to the diaphragm, and they
So it
play a
has the
major role in
discussion.
ensuring clear, sharp images. Okay? Then you have the diaphragm, also known as the iris diaphragm, inverse relationship, meaning a high resolving power or resolution means a low limit of resolution.
So basically here, this would be the alternative or the substitute rather for the pre-lab presentation during our in-class discussions. So this also has links to videos that are videos that are uploaded in YouTube. So later I'll show you. So this is the pre-lab PowerPoint with voiceover. Then for the PDF copy of the PowerPoint, let's go back to right. So we have also the link. So as you can see, this would be the content of the pre-lab PowerPoint video. So it contains all the slides of the video that I just showed you. Right. And then as you can see also you have the videos, and then if you click the image, that would automatically send you or bring you to the YouTube link or the YouTube video of that specific experiment or the specific video. So now to proceed with the experiment videos that we prepared. Let's go back. Right. So we have several videos prepared for this exercise. So you have different parts of the microscope, proper handling, how to use the OIO, and calibration. So as you can see, for every picture, you have a specific link that will show. And once you click this, this image, it will automatically bring you to the YouTube video. I'll just share the video. So let's share this. So it will bring you to this site. There. just to give you a sneak peek also of the other videos that we prepared. So the next one is proper handling. Just all right. The next one is proper handling of the microscope. So this one is how to use the oil immersion objective. Let's just stop this. Share the screen. There we go. So basically that's for the presentation of, I will not show this anymore. So this would this would be available anyway. So for the Microbiology Consortium members, so once you click the link, it will automatically bring you to the video. So yun lang po.
Thank you so much, Sir Daniel. Di po ba nakakatuwa yung background music? Habang dala ang microscope, mapapaindak at mapa-cha-cha-cha. Moving forward, for exercise number 2, we have another so-called Suki. She has served as speaker in 2 past webinars, and she will discuss the outline of their work entitled Aseptic Techniques in Pure Cultures, which she created with her co-faculty members at the University of the Philippines, Manila. So let me give the screen to Dr. Marilyn P. Balolong. Ma'am Len.
Ma'am Millet, unfortunately, Dr. Balolong will not be able to join us today because she is not feeling well. But we have already sent the video to Sir Bon. Can you just please play it?
Thank you, sir.
Good day. We wish to present Exercise 2 for Basic Microbiology, covering aseptic techniques and pure cultures. I am Marilen Parungaw Balolong from the University of the Philippines, Manila. Because microorganisms are everywhere, culture media must be sterilized before use. Once a sterile culture medium has been prepared, it is ready to receive an inoculum to start the cultivation process. This requires aseptic technique, which is a series of steps to prevent contamination during manipulations of cultures and sterile culture media. Perfecting the skill on aseptic technique is required for success in the microbiology laboratory, and it is one of the first methods that must be learned by a microbiologist in training. Airborne contaminants are the most common problem because the dust in the laboratory air contains microorganisms. So when containers are opened, they must be handled in such a way that contaminant-laden air does not enter. Aseptic transfer of a culture from one tube of medium to another is typically accomplished with an inoculating loop or needle that has previously been sterilized in a flame. First, loop is heated until red hot and cooled in air briefly. Then, the tube is uncapped and ran through the flame. Sample is then removed on sterile loop for transfer to a new sterile medium, and then the tube is reflamed and recapped. The loop is reheated before keeping. Cells from liquid cultures or sterilized culture media can also be transferred to sterile plates or fresh agar media using aseptic transfers. For more information about aseptic techniques, you can visit the link provided here. In relation to preventing contamination, the level of containment used to prevent accidental infections or accidental environmental contamination in clinical, research, and teaching laboratories must be adjusted to counter the biohazard potential of the organisms handled in the laboratory. Laboratories are then classified according to their containment potential or biosafety levels or BSL. They are designated as BSL-1, BSL-2, BSL-3, and BSL-4. BSL-1 laboratories are the lowest level of containment. Work can be done on an open bench with organisms that present a low risk of infection. These organisms are not pathogens in normal individuals and include organisms such as Bacillus subtilis. Workers should be protected by barrier protection, such as your laboratory coats and gloves. An example of a BSL-1 facility is a teaching laboratory that does not use pathogens. BSL-2 laboratories are designed to contain organisms that present a moderate risk of infection due to accidental ingestion, percutaneous injection, or exposure to mucous membranes via aerosols. Work with pathogens such as E. coli or Streptococcus pyogenes is done in a BSL-2 laboratory or sometimes at higher containment levels. Normal procedures may be performed on bench tops and must adhere to all BSL-1 precautions. An additional barrier protection such as face and eye protection, gloves, and laboratory coats or gowns must be used. Procedures that generate large volumes of organisms or that may generate aerosols must be done in a biosafety cabinet. Most microbiology research, clinical, or teaching laboratories now maintain BSL-2 containment standards. For more information about biosafety levels, you can visit the link provided here. Robert Koch, the German physician and microbiologist, is credited with founding medical microbiology and formulating his famous postulates. The postulates not only offer the means for linking the cause and effect of an infectious disease, but also stress the importance of a laboratory culture of the putative infectious agent. Once a culture medium has been prepared and made sterile to render it free of all life forms, organisms can be inoculated, and the culture can be incubated under conditions that will support growth. In a laboratory, inoculation will typically be with a pure culture. This is a culture containing only a single kind of microorganism. Picking an isolated colony and streaking it as the main method for obtaining pure cultures from samples containing several different organisms. To make a streak plate, to obtain pure cultures, the loop is sterilized and the loopful of inoculum is removed from the tube. A streak is then made and spread on a sterile agar plate. Following the initial streak, subsequent streaks are made at angles to it, the loop being re-sterilized between streaks. The appearance of a well-streaked plate after incubation is shown here. These are colonies of the bacterium Micrococcus luteus on a blood agar plate. It is from such well-isolated colonies that pure cultures can usually be obtained. For more information about the pure culture technique, you can again visit the link provided here. Microbiology is about diversity and evolution of microbial cells, about how different kinds of microorganisms arose and why. It is also about what microorganisms do in the world at large, in soils and waters, in the human body, and in animals and plants. One way or another, microorganisms affect and support all other forms of life, and thus microbiology can be considered the most fundamental of the biological sciences. Microorganisms are indeed everywhere, from our hand as a normal flora, or from common items such as our shoes, clothes, furniture, mobile phones, and the likes. They are then become fomites when they carry pathogenic microbes and serves as means of transmission. Microbes can be isolated in the laboratory using the capture plate method or the swab plate technique. For more information about this and to learn more about distinguishing normal flora from pathogens, you can visit the links that we have provided here. For the learning outcomes of exercise 2, determine the best practices for aseptic techniques to prevent contamination during transfers and apply aseptic techniques in preparation of pure cultures. Additionally, differentiate biosafety levels in handling microorganisms in the laboratory, enumerate common microorganisms that are part of the normal flora, and explain the role of fomites in disease transmission. To achieve these outcomes, here are the activities to be conducted. Activity 1 will be a synchronous activity via a video conferencing app. It may be Zoom, Teams, or Google Meet, or FB Rooms. I call it the Think, Discuss, Share. This is a non-graded assessment. Groups will be given photos of laboratory practices and they will discuss among themselves if they are correct or non-correct based on their knowledge of aseptic techniques. A group beadle will share the highlights of the group sharing, and afterward, the instructor will provide the synthesis. An alternative to this will be sending the images in the form of a module to the student for individual processing should there be challenges in connectivity. Consultation with the instructor can follow. Activity 2 will be a graded assessment. Should the instructor be deciding to check their knowledge about the basic concepts in aseptic techniques. This will be administered via the Google Doc or an LMS. You can click the link here for details. This quiz can be printed for those who are connectivity challenged and may be administered orally via FB or Viber call if it is necessary. For the next learning outcome on the application of aseptic techniques in preparation of pure cultures, a virtual 3D experiment will be provided using the link here. Their goal is to follow the directions and come up with a pure culture plate after the simulated incubation period. The instructor, during consultation, will also explain the troubleshooting related to the experiments. For the connectivity challenged, an art approach will be conducted. So here are some of the troubleshooting plates that can be explained and used by the instructor. And this will be the art approach for those who are connectivity challenged. The main goals of this activity is for the students to take a screen cap or a screenshot of the plated pure cultures. I wish to acknowledge my co-authors, Joel, EJ, and Aaron of the Department of Biology, University of the Philippines, Manila. And here are the links to the learning resources. Thank you very much, MCC, for this opportunity, and thank you everyone for listening.
Thank you so much also, Ma'am Len. Sana mapakinggan mo ang pasasalamat namin sa iyo at sa iyong buong team. Moving forward, let's go to exercise number 3, which is all about culture media preparation. For a rundown on this, Dr. Marilyn S. Sumayo and Ms. Maria Romela Angela C. Borromeo from the University of the Philippines, Visayas, have prepared a video teaser. Let's play the video, please.
Good day. We are from the Division of Biological Sciences, University of the Philippines, Visayas. And here we present the laboratory exercise that we developed for culture media preparation designed for remote learning. Here's the outline of the presentation, which is basically the same content in the laboratory exercise. For the introduction, the survival and growth of microorganisms depend on available nutrients and a favorable environment. Any nutrient material prepared for the growth and cultivation of microorganisms in the laboratory is called a culture medium. The different types of culture media are categorized according to chemical composition, principal function, and physical state or form. The culture medium must satisfactorily supply all the factors required by the microorganisms. Any medium must take into account the physical and chemical requirements for microbial growth. Culture media preparation is fundamental to cultivate microorganisms, obtain pure cultures, analyze microbial growth, and is used in many other microbiological applications. After accomplishing the exercise, the student can compare the different types of culture media in terms of form, function, and composition, and prepare culture media. Included in the exercise is a study guide. In order to understand the important points in culture media preparation, such as definition of terms, materials and equipment, and stepwise procedure, the student will read the culture media preparation manual and watch a video clip on culture media preparation in a laboratory setting. The study guide materials can be prepared by the instructor or can be found in other learning resources curated by the instructor.
Next, the student will proceed to the activity proper. Activity 1 is entitled Make Your Media at Home. The student will watch the video found in the link to have a clear visualization of the stepwise procedures in preparing culture media at home. After that, they will make their own culture media at home and submit the following outputs. So they are required to make a concept map or schematic diagram of the culture media preparation procedure based on the video, and provide proof of the actual conduct of making their own media via photographs or video presentation. Here are the materials and procedures in making their own media at home. For the activity 2, the student will examine if their prepared media is favorable for the growth of microorganisms. As an output, students will collect samples and inoculate on their prepared media following the materials and procedures presented in this slide. The results, including photographs, should be presented in the lab report following the worksheet provided. Shown to you in this page is page 1 of the sample laboratory worksheet. Notice that the flow and template of their output in activity 1 and 2. For page 2, students should answer study questions in a problem set. The problem set will be solved by filling in table 2. All student outputs will be assessed using assessment rubrics, which are included in the laboratory exercise manual. Lastly, here are the references used in developing this lab exercise. Thank you very much.
Maraming salamat sa team from UP Visayas. We're now halfway into the list at exercise number 4. My colleague in UPLB, Dr. Glazel Angelique Barredo, would show us a profile of her group's work entitled Microscopic Examination of Microorganisms. Ma'am Gla, the screen is yours.
Hello po, rinig na po ako.
Yes, ma'am.
Okay. Good morning po. So yung exercise 4 is the equivalent of exercises 2 and 3 in UPLB's micro laboratory. So for screen share. So for exercise 4, microscopic observation of living microorganisms, like for exercise 1, we prepared pre-lab video with voiceover, PDF copy of the PowerPoint, and the experiment videos. So for the pre-lab video, this is already uploaded in the G Drive. So all you have to do is click and you will be directed to the link. So for exercise 2, here is the prepared pre-lab video. So included here are the expected learning outcomes and the brief background for exercise 4 and the laboratory preparation itself and the expected results. So after finishing your experiment, color and other remarkable characteristics. So I will not show the whole video because this is worth like 16 minutes already. So it would take up all our time. So let me go back to the PDF. So for the PDF, you can also click the link and then you will be directed to the So can you see the PDF link?
Not yet, ma'am.
So hindi rin nakita yung video a while ago, right?
Yes po.
Ayan. So for the PDF link po muna, ito po yung link. So basically, this is the content also of the pre-lab video that I have shown a while ago. So meron ding links and activities that were prepared. So for the video, kita niyo po yung video ngayon?
Yes po.
Yan. So basically, yung pre-lab video, yan. 16 minutes siya, so hindi ko na ipapakita. But the information for exercise 4 is already here. So yeah. Then going back, aside from the pre-lab video and the PDF of the PowerPoint, we also have the experiment videos for the wet mount technique and the hanging drop technique. So like in exercise 1, you just have to click the video, then it would direct you to the YouTube link. So we also have an experiment video for the hanging drop. So you just have to click again the video. So for the hanging drop, this is the video that we prepared. So medyo mahaba din siya, so hindi ko na rin tatapusin. Then as mentioned, yung exercise 4 is equivalent to exercise 2 and 3 ng UPLB's micro. So Sir Daniel prepared the exercise 3 or the staining part. So I will just show quickly what he prepared. So for staining microorganisms, so same, we have the pre-lab video with voiceover. We also have the PDF copy that you can also access in the G Drive, and we have the experiment videos. So for the pre-lab video, just a moment. Sorry, mabagal ata ang internet. So basically, yun din yung video na nandun din sa G Drive. Then we also have the PDF. Same folder kung nasaan yung exercise 3 or the staining. Then for the experiment videos, we have one experiment video for this exercise. So aside from the YouTube links, the experiment videos were also uploaded sa G Drive. So all files for exercise 1 and exercise 4 are all together in one G Drive, aside from the YouTube links. So that would be all. Thank you.
Thank you also, Mangla and the UPLB team. Fifth presentation was developed by a member of the MCC board, Dr. Franco G. Teves from Mindanao State University, IIT. His module is all about fermentation. Sir Frank.
Welcome to the Basic Microbiology Virtual Lab, Introduction to Factors Affecting Microbial Growth. The rate of growth or death of a particular microbial species is influenced by a variety of physical factors in its environment, including temperature, osmotic pressure, pH, and oxygen concentration. In nature, where many species coexist, fluctuating environmental conditions cause dramatic population shifts due to the varying growth rates of different microorganisms. Every microbial species has a set of optimal conditions under which it flourishes. However, because the conditions in natural environments fluctuate widely, microbes have adapted tolerance to a range of environmental conditions. For example, many microbes have an optimum growth temperature of 30 degrees centigrade, but will grow, though slower, at 4 degrees centigrade. In the laboratory, where conditions can be controlled, it is possible to achieve optimal growth conditions for a given microorganism. For the intended learning outcomes of these activities, there are 4 that are listed. 1 is to be able to relate lectures on the effects of temperature, pH, oxygen, osmotic pressure, desiccation, and culture media components on microbial growth to controlled laboratory experiments. 2, to predict the general types of microorganisms that can be isolated from different environments. 3, to demonstrate how these lessons can have practical applications in a home. And 4th, to design a simple experiment to demonstrate the relationship between microbial growth and physical and chemical factors in the environment. In the virtual lab exercises, you will learn how to set up experiments demonstrating the effects of temperature, pH, oxygen concentration, and osmotic pressure on the growth of microorganisms. There are required readings on temperature requirements of microorganisms from basic microbiology textbooks such as Brock Biology of Microorganisms by Madigan and others, or Microbiology and Introduction by Tortora and others. Readings on oxygen requirements, pH requirements, water requirements, and lecture materials on microbial growth and anaerobic culture techniques. There are also recommended YouTube videos that you can watch in your own free time on bacterial temperature preferences, oxygen preferences, pH requirements, and environmental factors affecting microbial growth. See you all in our virtual lab from the MSU IIT Microbiology Group.
Maraming salamat, Sir Frank. I'll do a mea culpa first. The presentation is all about factors affecting microbial growth. Okay, moving forward, exercise number 6 will be outlined to us by a faculty member of the Cavite State University. Mr. Angelbert D. Cortez would now present, actually, I think he sent a video on this, a module on antimicrobial susceptibility testing. Video, please.
Good morning, everyone. In behalf of the Department of Biological Sciences of Cavite State University, I'm Angelbert Cortez, and I will be presenting to you a virtual experiment on antimicrobial susceptibility testing. The contents of the module include the introduction, which provides the background of the topic, the aims or what are the expectations from the students. We have also the procedure for the virtual learning or exercise. We have here the references that we use to create the virtual experiment, and of course, provided the activity worksheet, which will serve as the final output to the students after the exercise. For this exercise, we're going to use the diffusion method, particularly the Kirby-Bauer disk diffusion assay. So for the learning objectives, so we have the students should be able to follow the process for antimicrobial susceptibility testing through a virtual exercise. So since they cannot do the experiment hands-on, so they will just have to follow the process and how this activity works in the laboratory. Then we have also to determine the susceptibility of test organisms to different antimicrobial agents. So in the virtual laboratory, we have the 3 test organisms. So we have E. coli, Staph aureus, and Pseudomonas aeruginosa. These organisms were subjected to 13 antibiotics, or 12 antibiotics. And then we have here, the next one is to interpret the standard antimicrobial susceptibility test results based on the interpretive standards, whether the result is resistant, intermediate, or susceptible. For the procedure, we're going to use the Kirby-Bauer disk diffusion method. So here, this is the overview or the summary of the process of how the experiment is done in the laboratory. So the step-by-step process for this experiment was already placed in the module. But here, I'm just going to present to you the overview. So first is the preparation of inoculum and McFarland standard, at least 0.5 McFarland standard, which is approximately about 1.5 times 10 to the 8 CFU per ml. Of course, together with this inoculum, you have also to prepare the different concentrations of antibiotics. And then the inoculum is inoculated on the MHA or the Moeller-Hilton agar plate by creating a lawn. And then after 15 minutes, we have to deposit the antibiotic discs on the bacterial lawn, incubate, and then if there are zone of inhibitions, the students will be able to measure it and interpret data based on the standard. Let's proceed to the virtual measurement of inhibition zones and interpretation of results. So this is the virtual experiment that will be done by the students virtually. So here we have the virtual interactive bacteriology laboratory created by Dr. Cindy Griff Arvidsson of Michigan State University. We're going to use this virtual laboratory for assessment of students' performance. We just have to go with the link. So this is now the virtual laboratory that the students will do the hands-on using the virtual ruler in performing the experiment, particularly some measurement and interpretation of the results. Now for the activity worksheet, so typically they have to put their name, course and section, the date, and the score from the professor. So again, this table will gather the data from the virtual laboratory. And let's say for amoxicillin, the measurement is 24, they have to put 24. And again, this is millimeter. So the interpretation, let's say, is susceptible, so they have to put susceptible. So they have to record the data and so on and so forth. And then after that, we have here the evaluation, wherein students should state their specific insights on the results generated from the virtual laboratory. They have to explain the possible implications of the results. The next one is in key questions. We also provided questions that will assess the students' comprehension, let's say, understanding on the general concept of antimicrobial susceptibility testing. So we provided here questions, and professor can follow or can use the recommended questions, or they can also modify the questions. Now for the scoring of the activity worksheet, so we will give the students 15 points for the measurement values, so 5 points per test organism. So as you can see, there are 3 organisms, so 5 times 3 is 15. Now, the students or the professor should also consider that maybe the students could able to measure, like, for example, if the actual measurement is 24 and the measurement of the student is 23, so I think professor should consider that, because again, they're using or they are just measuring it virtually. So plus minus. Next is for evaluation. So we give them 5 points for their insights and opinions on the implications or the implications of the results. And the next one is the key questions. We have 6 questions. The number 1 to 5 questions, we will give them 3 points each. And for the number 6 question, we will give them 5 points, because you can see this is about designing experiment and how the students could utilize the available chemicals at home. And then the total score will be 40 points. So I think that's all for this experiment. For end, I would like to extend our gratitude to the Philippine Society for Microbiology for allowing us, the Department of Biological Sciences of Cavite State University, to create and present the virtual experiment on the antimicrobial susceptibility testing. Thank you.
Maraming salamat sa CABSU team.